All right, let's see if I can make this a little bit bigger. Put myself down here in the corner. All right, so this video is going to be about endosymbiosis and autogenesis, the two theories on eukaryote evolution. In other words, where did eukaryotes come from? The first is endosymbiosis, and the name tells you what it is. Osis is a process, endo means inside, and symbio tells you it's a symbiotic relationship. So it's a process of using a symbiotic relationship to get inside, in this case for one cell to get inside of another. And so what it says is that you had cell number 1, a large prokaryote, and cell number 2, a small prokaryote. And normally what would happen is cell number 1 would engulf number 2, bring it inside, and digest it using the enzymes. But what they said happened instead is that cell number 2 made cell number 1 a deal it couldn't pass up and said, hey, if you won't kill me and eat me, I'll do a job for you. And so they agreed to help each other out. Cell number 2 gets a nice, safe place to live. Cell number 1 gets the benefit of energy or food or whatever. So they believe that this is how the mitochondria and chloroplast arose. Some people fight and say this is how all of the organelles arose, but most people agree that at least the mitochondria and chloroplast arose this way. Mitochondria being first, because when this happened, it created the eukaryote, and this became the mitochondria, and all eukaryotes have a mitochondria, and then those lines split, and another one of the lines did it again, and this time that organelle or that prokaryote became the chloroplast. The other group kept just living with the mitochondria, and that's you and me with no chloroplast. All right, so what is the evidence for this? So the evidence supporting this theory is that mitochondria and chloroplast are in similar size and shape to prokaryotes. So when you measure prokaryotes and you look at their shape, and then you look at a mitochondria and a chloroplast and their size and shape, they're very similar. Eukaryotes are much larger. Their DNA is similar. So prokaryotes have a single circular chromosome. Guess what? So do mitochondria and chloroplast. The steps to transcription. When you do transcription as a eukaryote, you as in actual you, there are a bunch of steps to it. Prokaryotes do it in a much more simple fashion, and when you look at mitochondria and chloroplast, they do that simpler version of transcription as well. We'll learn more about that in Unit 6. Their genetic code is slightly different. So in us, UGA is a stop codon, but in prokaryotes and mitochondria and chloroplast, it is not. It actually codes for an amino acid. They divide by fission. Fission is division, but we don't do fission. We do mitosis or meiosis. And so when we look at mitochondria and chloroplast, they do fission. The ribosomes that you see in prokaryotes are smaller than the ribosomes that you see in our cells. But when you look at the ribosomes that exist in mitochondria and chloroplast, first of all, there are ribosomes, so that suggests it was a cell. But they are about the same size as the ones found in prokaryotes, much smaller than the ones found in us. And then lastly, the fact that they have a double membrane. So the double membrane suggests that they had their own membrane, and then, and I didn't show it, whoopsie, then when they were engulfed, this cell membrane that wrapped around them and formed a vacuole or vesicle around them would be their outer membrane. Okay, and it's not truly a vacuole or vesicle because that was a prokaryote. But anyway, so that's for that double membrane. Now, what is the evidence or the criticisms about why this may not be an accurate theory? The first thing is the amount of energy. Prokaryotes did not have access to a lot of energy because they were so small they could only take in so much food, and this would have required a lot of energy. And so that's one complaint or one criticism. Another is that if the nucleus was formed through endosymbiosis, how on earth did it get, in our case, 46 linear chromosomes? How did we get from one circular chromosome, if it was indeed a prokaryote, to 46 linear? So we'll call that nuclear DNA. Another is that bacteria love anaerobic environments. That's supposed to be a heart. But the cytoplasm tends to be aerobic. So how, if this was a bacteria, how would it have survived in this aerobic environment in here when it came in if it was an anaerobic bacteria? Another thing is, how did it convince cell number 1, who could have gotten a meal out of it, so we'll call that competition or predation, how did it lose out to symbiosis? I mean, whatever number 2 was offering had to be really good for cell number 1 to say, yeah, I don't think I'll eat you. I think I'll keep you around, because cell number 1 could have gotten energy from that. So there had to be something good number 2 was offering for symbiosis to win out. And then the last one is that there had to have been lots of steps. This is not something that would have just one and done happened. It had to have happened over a long period of time and with a lot of steps. And so that's a complaint with evolution in general, is that it is so multi-step. How did it ever happen perfectly? That's not really the right word, but think of all the imperfect steps that had to have happened where cells would have died before we got to the versions we have now that are functional. So that's endosymbiosis in a nutshell, the supports, the criticisms. Autogenesis will be a little bit faster. Autogenesis is where, so auto means self, genesis means creation, and so it is the creation of self, or the creation of things for itself. So what they think happened is that maybe the cell folded in and kept folding in. Now let me think of how I want to show that, until these two pieces came together and closed here, and then that broke free and became an organelle. Okay, so that is autogenesis because there's no second cell. It's all happening with itself, and it is creating more structures for itself. The support for this is that all of those organelles that have a single membrane, your rough ER, your smooth ER, your Golgi, your nucleus, your peroxisome, lysosome, vesicle, vacuole, all of those that have a single membrane, this makes sense. Okay? Because when you fold in on yourself and you pinch off, that is going to just be a single membrane. It's not going to be a double membrane like here where you're engulfing another cell. So for the single membrane organelles, this makes a lot of sense. It works, you could say. All right, criticisms, and these lists are a lot shorter for autogenesis, is that what about the ones that have a double membrane, like the chloroplast and the mitochondria? Where'd they get that double membrane? Some scientists suggest that maybe they did this and then they folded in on themselves again. They could have. That's definitely a possibility. It's just not super likely. And then again, one of the same criticisms that the endosymbiotic theory had is that this would have had to have happened in lots of steps. So it would have had to have happened again and again and again. Once for the nucleus, once for the ER, then that would have had to split into smooth and rough, and for the Golgi. And it just would have had to happen a lot of times over and over and over again. Could have. Definitely could have happened, but it just makes it more unlikely. So those are your two theories, your support and criticisms of each, and I hope that helps. If you have any further questions, let me know.